Now, without further ado, I would like to invite Professor Dr. Muhammad Jamil bin Yaakob to introduce our speaker today.
Thank you, Ms. Shafarina. Assalamualaikum warahmatullahi ta'ala wabarakatuh. A warm welcome to everyone. Thank you for joining our session today. This is the second day of the Medical and Health Sciences Conference organized by UniSHAMS. Without further ado, let me introduce you to our speakers today. Dr. Masita Arip. Dr. Masita Arip is head and consultant pathologist at Allergy, the Allergy and Immunology Research Center, Institute for Medical Research, National Institute of Health, Ministry of Health, Malaysia. She was trained as an undergraduate in Universiti Kebangsaan Malaysia in 1999 and subsequently obtained her master in pathology, in microbiology and immunology at UKM in 2008. She is currently a consultant pathologist for medical microbiology and immunology and head of Allergy and Immunology Research Center, Institute for Medical Research, National Institute of Health, Shah, Selangor. She provides consultations to Ministry of Health and non-Ministry of Health organization for immunology, medical microbiology, and also the area of vaccine. She's also the Dean of Diploma Medical Microbiology course in IMR under Southeast Asia Ministry of Education Tropical Medicine program. Dr. Masita involved in more than 40 researches focusing on allergy, autoimmune, transplant, immunology, tropical infectious diseases, and vaccine. She has published more than 40 original articles and proceedings in different types of journals, locally and overseas. She is also responsible in setting up Malaysian Stem Cells Registry in the year 2000, which is a database for healthy Malaysian volunteers who can donate blood stem cells to patients with blood disorders. She is also involved in lab quality management system, and she is the quality managers of QMS ISO 15189 in IMR since 2016. All the 11 diagnostic labs in IMR had been accredited with MS ISO 51809 since 2014. She's actively participated in as lead assessor and technical assessor for Scheme Akreditasi Makmal Malaysia, organized by Jabatan Standard Malaysia. I think she's the right person today to talk on COVID-19 vaccine program in Malaysia. So without further ado, let me invite Dr. Masita Arip to deliver her talk. Dr. Masita, it's yours.
Apologize for the technical hiccup just now. So the title that I will share today for my presentation, titled COVID-19 Vaccine Program in Malaysia. As mentioned by Prof. Dr. Jamil just now, I'm currently working at Institute of Medical Research, IMR, located at National Institute of Health, Setia Alam, Shah. Next. So the outlines of my presentation today, I will talk about the COVID-19 vaccine program. Secondly, about the vaccine efficacy, vaccine effectiveness, WHO SARS-CoV-2 variant working definition, COVID-19 vaccine enrollment in Malaysia, and I will discuss more about, I will focus more towards the 3 vaccines currently used in Malaysia for the vaccine efficacy, the Pfizer, AstraZeneca, and also Sinovac. Next. About the COVID-19 vaccine program. Next. The National COVID-19 Immunization Program Guidebook launched at 16 February 2021, where it outlines the National Strategic Plan of National COVID-19 Immunization Program to run smoothly, safely, effectively in an orderly manner. So this multi-approach, multi-ministry also, between Ministry of Health, MOH, Ministry of Foreign Affairs, and the Ministry of Science, Technology and Innovation, mostly. This to ensure that we can secure access to vaccine procurement and reduce dependence on a single source of supply, negotiation with COVID-19 vaccine manufacturers, value add to the development of the national supply of vaccines. For your information, during this discussion, we started the discussion actually since last year, where we need to negotiate with many the vaccine manufacturers and also government, for example, China government and so on. So all these negotiation to ensure that we have multiple source of vaccines. Next. So in this guidebook also, we already stated the government strategy is to ensure as many residents in Malaysia as possible will receive the vaccine to save lives in the fastest possible time. And the vaccine will be provided for free to all citizens and non-citizens of Malaysia. Why? Because we want all Malaysian population can prevent it from getting the COVID-19 infection. So this is what our Prime Minister wish during the statement in the National COVID-19 Immunization Program book. Next. So government already secured 79.5 million doses of COVID-19 vaccines through the COVAX facility and advanced purchases from 5 vaccine manufacturers. This is updated until June 2021. So what is COVAX? COVAX is actually a COVID-19 vaccine global access facility. Is a global initiative launched by Global Alliance for Vaccine and Immunization, Gavi, the Coalition for Epidemic Preparedness Innovation, CEPI, and the World Health Organization, WHO, to provide equitable access globally for diagnostics, treatment, and vaccines. So we also have bilateral agreement between Malaysia and China last year in November. So that has a priority access to COVID-19 vaccine. As stated here, this diagram showing the one of the information about the supply of COVID-19 vaccines that have been acquired by Malaysia. And during the preparation of this book, they already put here, we will secure the doses of vaccine will be 66.7 million during the time that this book launched during February. But now, as you can see, the government already increased the securing of the vaccines, not only 66.7 million, now we already have secured 79.5 million. Why? Because we need to remember our population, sometimes not all people can receive one type of vaccine. Sometimes they need to give another type of vaccine because of allergy or so whatsoever. So that's why we need more than the plan. And as you can see, our coverage for vaccine actually more than 100%. More than 100%. Okay, next. So in order to make sure that our vaccines really effective and safe, we have our Pharmaceutical Regulatory Agency, the National Pharmaceutical Regulatory Agency, NPRA, is actually under Ministry of Health, is the body responsible for evaluating the vaccines to be registered in Malaysia. Secondly, the Drug Control Authority, after the NPRA already evaluate the vaccines is safe and effective, the Drug Control Authority will approve the use of vaccines based on the results of NPRA evaluation. And the Ministry of Health, MOH, will monitor the adverse events following immunization after the vaccine is administered in Malaysia. So this AEFI, adverse event following immunization, means if anything happen, whether you have severe reaction or just a mild reaction can be just maybe vomiting or maybe just itchiness. That one can considered mild. What about the AEFI, which is severe, can be difficulty breathing, asthma, even fainted. That one considered serious. So this AEFI will be reported either by the healthcare professionals, the doctors in the hospital or clinic, or from even the pharmaceutical companies, and can also from us, from our public. They can report all this reaction to the system, what we call the National ADR Monitoring Center, NPRA. So if you go to website of NPRA, there will be a column to report any reaction after receive the vaccines. And then from this information, they will revise into the database, and there will be a meeting to discuss about the AEFI. And we must submit all this information to the WHO also, yeah? Not only kept in Malaysia. Next. So COVID-19 vaccination policy is a voluntary free of charge for those living in Malaysia, aged currently 18 years and older, and to ensure at least 80% of Malaysian adult population receive vaccine by February 2022. So the interval between first and second dose of the 3 vaccines currently in Malaysia for Pfizer, 21 days, AstraZeneca 28 days, and Sinovac 14 days. So the distribution of vaccine is stages to nationwide based on supplies and deliveries from vaccine manufacturers. That's why at the initial phase, during the phase one actually for the maybe can next slide. For phase one, actually for frontliners, workers, medical and non-medical staff, from February to April, the quantity of the vaccine during that time also very limited. That's why they've only focused to this group. And then for phase two, starting from April until now, for the high risk group, senior citizen, the number of vaccines already increased a lot, and especially now, we already can give to the adults aged 18 years and above. And hopefully, from what we understand from our Minister Mosti, he said that if possible to give it by before October, hopefully because before this we thought that we can achieve the 80% by February. Maybe it can be earlier. Next. So now I want to actually share what is the meaning of vaccine efficacy. Next. Approximately COVID-19 vaccines are at various stages. 96 COVID-19 vaccines are at various stages of clinical development. So the vaccine efficacy measures a vaccine's protections against a disease or pathogen in a vaccine trial. This is very important. The word here, vaccine trial. When we said about vaccine efficacy, usually in the vaccine clinical trial. And if a vaccine has, for example, 70% efficacy, it means a person vaccinated in a clinical trial is around 2/3 less likely to develop the disease than someone in the trial who didn't get the vaccine. So that's the meaning of the 70% efficacy, means that 70% of the people will be protected from getting the disease. Okay, next. What about vaccine effectiveness? Next. Vaccine effectiveness measures when the vaccine is given outside the clinical trial. And compared with efficacy, effectiveness takes into account all the complexities of the real world outside of the controlled clinical trial setting, because usually in the clinical trial we have the criteria for inclusion criteria for the receiving subjects, but for vaccine effectiveness we just measures the effectiveness by all the population. We didn't control the inclusion or exclusion criteria. Okay, next. So is there a standard vaccine efficacy threshold, and should a lower level be concerned? At the moment, there is no single standard vaccine efficacy threshold applied to all vaccines. Any decision to use a vaccine or drug always involve weighing the benefit against the risk. So the 50% efficacy threshold set for COVID-19 vaccine is because COVID-19 was deemed such a severe disease that is, if a vaccine is only 50% effective, it's still worth using. And fortunately, the emerging data on COVID-19 vaccine suggests that the vaccines are very safe with high efficacy, at least against some of the variants. Okay, next. I would like also to share about the WHO SARS-CoV-2 variant working definition about the COVID-19 mutation. Okay, next. So this WHO SARS-CoV-2 variant working definition last updated 6 July 2021. They categorize the mutation of the virus into 2 categories, the variant of interest and the variant of concern. So what is the meaning of variant of interest? Variant of interest means a SARS-CoV-2 variant with genetic changes that are predicted or known to affect virus characteristics such as transmissibility, disease severity, immune escape, diagnostic or therapeutic escape, and identified to cause significant community transmission or multiple COVID-19 clusters in multiple countries. And for variant of concern, a SARS-CoV-2 variant that meets the definition of variant of interest and through a comparative assessment has been demonstrated to be associated with one or more of the following change at the degree of global public health significance. First, increase in transmissibility or detrimental change in COVID-19 epidemiology, or increase in virulence or change in clinical disease presentation, or decrease in effectiveness of public health and social measures or available diagnostic vaccines and therapeutics. In other words, variant of concerns means dangerous. So we are very worried about the variant of concern if available in our country. As compared to variant of interest, we are not. It's not really too critical. But from this variant of interest, sometimes it can become the variant of concern if either one of the three characteristics like increased transmissibility, increase in virulence, or decreased effectiveness. From BOI also can become BOC. Next. Okay, and before this, the VOC, the variant of concern, they usually state the country who started the finding the variant, the mutation. But after that, they change. The UN Health Agencies uses letters of the Greek alphabet to avoid stigmatizing nations where they were first detected. So now we have Greek letters for the variant of concern or variant of interest. For example, for VOC now, for alpha, that if there is mutation B117, first detected in UK in December 18, 2020. And then beta, the mutation B1351, South Africa. Gamma, P1 Brazil, January 2021, and Delta, B1617.2 India, May 11, 2021. So this the four variant of concern that we really afraid that can cause difficult to contain it. So for the variant of interest, we have epsilon, zeta, eta, theta, iota, and kappa. And recently, kappa in India, they also news in India now. We are quite worried about the presentation of this kappa, B1617.1. Next. Okay, about the COVID-19 vaccine enrollment in Malaysia now, the status. Next. So at the moment, we have 3 vaccines. Pfizer, BioNTech, AstraZeneca, and Sinovac. Okay, next. So as you can see, the latest status updates 1st August 2021. How many already our Malaysian population for adult population 60.9% already received the first dose, and 29.7% already received the second dose. In terms of population, because population here must remember we include also the children. So in terms of population, 43.6% already received the first dose. So in other words, what we target just now, we target that in Malaysian population at least 80%, right? 80% must be vaccinated. So now we already like for first dose, like 50% already of our population, 43.6%. And for the 2 doses, 21.3% among the population. So we hope, that's why we hope that within these 2 months we can achieve 80% for our population receive the vaccine. And in terms of the states here, I think the highest is Labuan, 95.1% receive the vaccines. According to here, the population 65.4%. I think in terms of population, in terms of state, still quite far away. So maybe after this, we hope that within these 2 months, all these states also can achieve 80%. Okay, next. So now I will talk about the type of COVID-19 vaccines, efficacy for the 3 vaccines, Pfizer, AstraZeneca, and Sinovac. Okay, next. Now we go to the first one, mRNA vaccine COVID-19 efficacy. Next. So what is mRNA vaccine? mRNA vaccines are made using a new technology. During this COVID, we actually introduced a new technology, which is mRNA vaccine that can be used. Before this, only research-based. So in the case of COVID-19 uses a nanoparticle to deliver the genetic instruction for human cells to make the SARS-CoV-2 spike protein. This priming the immune system to attack the SARS-CoV-2 virus if later infect the body. So as you can see here in the virus, SARS-CoV-2 virus, there is spike protein, right? This spike protein, the crown shape like. So from this spike protein, we can have the genetic sequence. So from this genetic sequence, we can we can build the messenger RNA that contains instruction to make the spike protein from a DNA template in the laboratory. But this mRNA is not really stable, so they need to, because it can easily digest by the enzyme inside the cells. So we need to put inside the nanolipid particle to ensure that the mRNA can just enter the cells without degrade. So this lipid nanoparticle, the mRNA, is encapsulated in the lipid nanoparticle, which stabilizes and protects the mRNA and helps it to enter the host cells. So for example, in Pfizer now, we have this mRNA inside the nanolipid particle, and we inject the vaccine inside our body, and then they will go to the what called the antigen presentation. The nanoparticle are picked up by the antigen presenting cells, APC. So APC, antigen presenting cells, usually what? Dendritic cells, all the macrophage. The mRNA is stripped by the cells machinery to produce spike protein and then degraded it, and it does not enter the nucleus. Very important. The mRNA does not enter the nucleus. So some spike protein are broken down into short peptides, which activates all the T lymphocytes, all these T lymphocytes, and from these, some of the T lymphocytes, the T helper cells, can be also activate the cytotoxic T cells, the B cells, and these B cells, some become the plasma B cells. So these plasma B cells, we can produce the antibody, antibody that already specific to COVID. So some of the B cells also can become the memory B cells. Memory B cells, memory T cells that recognize the specific spike protein from the virus. So these memory B cells and memory T cells very important, because if there is infection, if there is SARS-CoV-2 enter to our body, so they will activate the memory B cells, will activate the plasma B cells. Memory T cells will activate the cytotoxic T cells. So they will actually work for plasma B cells to produce the antibodies. For the cytotoxic T cells, they will kill the cells that contain the virus. So that's how the immunology system, in terms of the immune response, how our vaccine works in our body. Next. Okay, next. Next. Okay, so all right. So vaccine efficacy mRNA for Pfizer BioNTech. For phase 2 and 3 study results published 31st December 2020 suggest that 2-dose regimen of BioNTech will give 94.6% protection against symptomatic COVID-19, at least 7 days following the second dose. So since then, there is a real-world studies have been published. Data from observational study of 7,000 healthcare workers in Israel suggest that vaccine efficacy against symptomatic COVID-19 is 85% from 15 to 28 days after the first dose. And further data from Israel mass vaccination program, vaccine efficacy at days 14 to 20 after the first dose and 7 or more days after the second dose as followed. For documented infection, 46%, and 92% for symptomatic COVID-19, 94%, for hospitalization, 74%, and for severe disease, 62 and 92% respectively. In terms, this is how the vaccine effectiveness in Israel. So the estimated vaccine effectiveness in preventing death from COVID-19 is 72%. So these are the studies from Israel. Next. Sorry. Okay. What about Pfizer? Is it effective towards the variant? So we already know just now, right? Alpha variant from UK. So this Alpha variant, observational data from the vaccine rollout in Qatar, where 265, 410 people have received 2 doses of Pfizer, So they published in 5th May 2021 suggests vaccine efficacy is 89%, 89.5% after the second dose, 14 or more days after the second dose. And vaccine efficacy against severe, critical or fatal disease caused either by alpha or beta are high, 97.4%. So this national level data also from Israel vaccination program confirmed that the vaccine is highly effective against the alpha variant, because from their study it showed that 95% of their tested specimens showed a reactive towards this SARS-CoV-2 variant, alpha variant. Okay, next. What about the beta variant? Beta variant actually the mutation B1351 first detected in South Africa. So in a small laboratory using vaccine sera suggests 10.34 reduction in antibodies that neutralize this virus. And however, analysis of 800 participants, phase 3 trial announced by the company's 1st April 2021 shows 9 cases of COVID-19 in the placebo group, 6 of which were due to beta and 9 in the vaccine group, suggesting vaccine efficacy of 100%. And another observational data from the vaccine rollout in Qatar, where 200, this is again similar with the one I mentioned earlier for alpha and beta, showing the vaccine efficacy 97.4%. Okay, next. What about Pfizer against the gamma? The gamma actually P1 mutation first detected in Japan or Brazil. A small laboratory study using vaccine sera suggests roughly equivalent neutralization of this variant, although 2 other laboratory studies published suggest antibody titers were reduced by 2.6-fold and 14-fold. In other words, although it's stated here antibody titers were reduced, but actually the antibody still can be detected. As you still remember during my explanation just now, we still have the memory B cells and memory T cells, and these memory B cells and memory T cells is very difficult to measure in our blood, because it's very specialized test. So people can only easily measure the antibody titers. Okay, next. So now, what about vaccine efficacy towards our latest variant, Delta, B1617.2, which is first detected in India? A study using vaccine sera from 250 participants after 2 doses suggests neutralizing antibody titers against the Delta variant reduced 5.8-fold compared with wild-type virus, but are still able to neutralize the virus to some extent. And another study, an updated analysis including 14,019 asymptomatic cases infected with Delta variant was published on 14 June. The analysis suggests vaccine efficacy against admission to hospital with Delta variant is 94% after 1 dose and 96% after 2 doses. Another study, Scottish study published 14 June also in Lancet, included 19,543 cases with and 7,723 with the Delta variant, of whom the 377 were admitted to hospital. The vaccine offered 79% protection against the Delta variant and 92% against the Alpha variant. So in other words, although we are really afraid with the Delta variant now, especially in Malaysia, so this study suggests that Pfizer still can produce and the antibody which can prevent severe cases. For example, in this publication. Okay, next. Now we go to the second vaccine, adenoviral vector vaccine COVID-19. Adenoviral vector actually AstraZeneca. Okay, next. So this adenoviral vector vaccine is also a new technology which uses a common cold virus to deliver the genetic instruction for human cells to make the SARS-CoV-2 spike. Here, the common cold virus is already like modified to ensure that they are not causing any illness to us, but at least it can prime our immune system, priming the immune system to attack the SARS-CoV-2 virus if later infect in the body. So again, from this diagram here, the spike protein again, the spike protein, we take the isolated gene from the spike protein, and this isolated gene from the spike protein containing the information of the spike protein is synthesized in the laboratory by reverse transcriptase into the complementary DNA, and we inserted inside the vector. So a human or simian adenovirus, which usually causes the common cold, is genetically altered to be non-replicating to carry the synthesized spike protein genes. So we insert these viral vector vaccines, and the viruses are picked up. The vaccines, which contain the adenoviral vector, will pick up by the antigen presenting cells, and the viral DNA is transcribed and translated by the cell machine to produce spike protein. Again, the same mechanism after this will be followed like that, like the mRNA also. The body will stimulate the T helper cells, the helper cells also stimulate the cytotoxicity cells, the B cells, the memory B cells, the plasma cell, B cells to produce antibody and so on. So these are the principle how the viral vector vaccines act in our body. Next. So what are the vaccine efficacy for AstraZeneca? Data released as a preprint in the Lancet, 2nd February 2021 suggests vaccine efficacy against symptomatic infection is 76% after single dose and increasing to 82.4% after a second dose and administered 12 weeks later. And there were no hospitalization or deaths in the vaccine group. So AstraZeneca announced result of study in the US, Peru and Chile, which involved 32,149 participants, suggests vaccine efficacy is 76% against symptomatic disease and 100% against severe or critical disease, with 2 doses given 4 weeks apart. Next. So data published on 1st July suggests that 2 doses of AstraZeneca are estimated to provide 94% against death from COVID-19 in people aged 65 and over. And a study published in 28 June 2021 suggests that longer delay before the second dose of vaccine leads to an increased antibody titer after the second dose. And a third booster dose given 44 to 45 weeks after the second dose induces antibodies to a level that correlates with high efficacy after second dose and boosts T-cell response. Of course, when we give multiple doses of vaccine, we will again prime our immune system and will produce more antibodies in our body. Next. So what about the effectiveness against the efficacy, vaccine efficacy of AstraZeneca against the new COVID-19 variant? For Alpha, the real data in UK vaccination program suggests high vaccine efficacy against the UK variant. And one study published in Lancet stated that neutralizing antibody titers generated by the vaccine were 9-fold lower for the UK variant, and vaccine efficacy against symptomatic cases were similar to Alpha and non-Alpha lineage. So here just to mention that the neutralizing antibody titers is slightly lower for the UK variant. For AstraZeneca, for Beta, a small laboratory study using vaccine suggests 12.4-fold reduction in antibodies that neutralize this variant. And another study in New England Journal suggests that the 2 doses regimen has an efficacy of only 10.4% against mild to moderate COVID-19 in almost 1,500 HIV negative young adults. So this is about the beta for AstraZeneca. Next. What about the gamma? Only one study, a small laboratory study using vaccine suggests antibody titers were reduced by 2.9-fold against this variant. And for Delta, there is a published data 22nd May, which included 1,054 people with having Delta variant, suggests that the AstraZeneca has an efficacy 59.8% against symptomatic Delta variant, as compared 66% against the Alpha variant. And another study here involving 14,019 symptomatic cases against the Delta, the vaccine efficacy with Delta variant is 71% after 1 dose and 92% after 2 doses of AstraZeneca. And similarly for the Scottish study, if you can notice, the UK and Scottish, they study both the AstraZeneca and Pfizer. That's why the results here, you can see that the vaccine offered for AstraZeneca for protection against the Delta variant is 60%. Next. Now we go to the inactivated virus vaccine, COVID-19 efficacy. Next. So these inactivated whole virus vaccines are made using an old technology. So this is very what we call known technology, which delivers as an activated version of SARS-CoV-2 virus directly into the body along with an adjuvant, which can prime the immune system. So how they do this? First, we must grow the virus in the cell culture. SARS-CoV-2 is grown in a large amount in the cell culture, and from these viruses that we already grow in the lab, the viruses are then treated in a way that disable the genes. So this process of inactivation can be either by heating certain temperatures or by chemicals or by radiation. We can kill the virus. So after that, because the virus is already killed, so the immune system sometimes is not really good in terms of priming our immune system. So we need something to boost the immune system. So that's why they add the adjuvant, usually the aluminium, to boost the immune system using the inactivated vaccine. So once we inject the inactivated vaccine to our body against, will be picked up by the immune cells called the antigen presenting cells, break up the virus and present the peptides to the all the immune cells. The helper cells. The helper cells will stimulate the cytotoxic T cells, the B cells, and so on. Similarly, with the AstraZeneca and also Pfizer, to ensure that our body already have all this immune system in our body, the memory B cell, T cells, and all the antibodies. Next. So the vaccine efficacy is inactivated for in our country, Sinovac. So this vaccine efficacy study for Sinovac, a large phase 3 trial in Brazil showed that the 2 doses administered at 14 days apart had an efficacy of 51% against symptomatic patient, and 100% against severe COVID-19 patient, and 100% against hospitalization. So this is the vaccine efficacy for Sinovac. And the preliminary and not yet peer-reviewed findings in Chile involving 2.5 million individuals age 16 over received 2 doses, and 2.1 million that received 1 dose suggest effectiveness against symptomatic starting 14 days after the second dose is 67% against the SARS-CoV-2 infection, 85% against the hospitalization, and 80% against death. Okay, next. So what about the Sinovac? What is the vaccine efficacy from Sinovac to the new COVID-19 variant? For Gamma 1, P1, the preliminary findings from another vaccine effectiveness study in Brazil, the effectiveness to be 49.6% after the first dose for P1 variant. And vaccine effectiveness assessed in cohort of healthcare workers in Sao Paulo, Brazil was 50.7%. And mainly in this Brazil, mainly P1 variant. Okay, next. So in conclusion, from all this information, the assessment of the efficacy of vaccine is complex for many diseases, but particularly so in the case of SARS-CoV-2, where the fundamental understanding of infection is evolving. Why evolving? We keep seeing the mutation in SARS-CoV-2. So it is probable that there will not be a single vaccine winner. Diverse platforms and technologies can offer different strengths and be relevant in distinct epidemiological contexts. However, collaboration and standardized approaches for assessing different efficacy endpoints will be important to allow meaningful comparison and ensure that the most effective candidates are deployed. And following deployment, well-supported pharmacovigilance studies should be established to ensure the ongoing evaluation of vaccine safety. Next. Just to give some inspiring quotes by the Amina Muhammad, Deputy Secretary General of the United Nations. What she said, we are encouraged by the rollout of safe and effective vaccines, but the truth is simple. No person is safe until all, everyone, everywhere are safe. And no country is safe until all countries are safe. Only by working together can we ensure that no one is left behind. Only by working together can we recover better and build a world where everyone thrives in peace, dignity, and equality on a healthy planet. It is possible together. So I hope with this quote, all of us must come together, take the vaccines and follow the SOPs in order to break the chain of COVID-19. Next. Next. Before, sorry. Okay. I would like to actually introduce the National Institute of Health, NIH, under the Ministry of Health. This is a new building, new complex in Setia Alam, Shah Alam, where we have all the management office, all the research institute under the Ministry of Health. IHM means Institute Health Institute Health Management. IHSR, Institute Health System Research. IHBR, Institute Health Behavioral Research. ICR, Institute Clinical Research. IKU, Institute Public Health. IMR, Institute Medical Research. And we also have the residency, we also have hotel in this complex. So if there is no more MCO, maybe I would like to invite all the lecturers and students and come and visit us in NIH at Setia Alam Shah Alam. For that, I thank you for all the attention given by the participants. Thank you very much.
Masita, for the invitation for us to visit the IMR, and thank you very much for your very informative lecture, which I think have enlightened us a lot on the efficacy, as well as the effectiveness of vaccine, the 3 vaccine that you mentioned. I have one question to you. Now, what about regarding the person who have already been infected by COVID-19, and what kind of immune response as well as the efficacy of the vaccine that were given after that or later?
Actually, for the people who already recovered, they will have the antibody also in their body. They also have the memory B cells and T cells, but the duration of these antibodies varies among individuals. Some people, the antibodies can last for 3 months. Some people 6 months. Some people longer than 6 months. So once we inject the person who already recovered with COVID-19, we actually introduce some sort of booster effect to the patient. In other words, they can get multiple larger amounts of antibodies or immune cells after that. So to answer that also, we in IMR we have both studies, Prof. Jamil, the longitudinal study also conducted by myself and my colleague in IMR, where we follow up all the recovered patient. We follow up for time frame, 3 months after infection, 6 months after infection, 12 months and 18 months, and we measure their immune system, the antibody cells, the neutralizing antibodies. And at the moment, we already collected until up to 1 year of samples. So from these preliminary findings, we can see that after 3 months, the majority, nearly 90% of them having the antibodies after 3 months, still have the antibodies. And then after 6 months, slightly reduced, less than 90 around 80%. And for the 1 year, we are still not analyzed yet because still have few people not completed the 1 year sample. So that one for the longitudinal study. And then we already have the what we call post-vaccination immunity study also. That one also under my team also in IMR, where we monitor the effectiveness and we want to compare the one that already recovered using single dose. How is the immune response? The one that you asked just now, right? How the immune response as compared to the one who received the first dose, which don't have any infection before that. So what we call as the immunity response among the recovered single dose vaccines among the recovered patient. We want to see, but because overseas already have data, but because the unique in our Malaysia, because we have 3 vaccines, right? Pfizer, AstraZeneca, and Sinovac. So we want to measure all these 3 vaccines, the vaccine efficacy or the vaccine effectiveness among the 3 vaccines after among the recovered patient. So I hope I answer your question, Prof.
Yeah, just from your preliminary data, do you think that we need another booster after 1 year or what?
Of course, Prof. Of course. It's good that we have more protection. You know why? Because even now, if you notice, quite many people also become infected even after 2 doses. So we must remember the one that already infected with the 2 doses, because that's a new variant. So for initial part, our body maybe cannot recognize the virus from this new variant, because our vaccine all from the wild type, the original virus, our antibody towards the original virus. But when this variant just come and see us, for initial part, our body cannot recognize until they process. Once it's processed, remember just now, the antigen presenting cells, the APCs process the virus, then our body recognize, oh, this is actually the peptide, the spike peptide. So, or the antibody from the vaccine can just kill. That's why all patients who already vaccinated, even they get infected, they are not become severe because of the antibodies can kill the virus, can attack the virus.
Yeah, but
So,
by 1 year, perhaps we already have herd immunity. Do you think still
uh,
possible
but
with herd
but
immunity getting
yeah,
another booster?
yeah. Herd immunity agreed that we can have a herd immunity, but again, I think for this, because this is the challenge for this virus, Prof. This virus keep mutate. That's our problem now. The virus keep mutate compared to other diseases. For example, diphtheria, pertussis. That one bacteria. They are not easily mutate. So that's why our childhood immunization just for childhood. No need for adult to get the vaccination because our herd immunity for childhood virus, bacteria, organism is already covered. But for SARS-CoV-2, if you notice, we are still struggling. Not only our country, okay, all over the world, struggling with this COVID, SARS-CoV-2 because they are keep changing, keep mutating. So in order to prevent all this complication, or all this hospitalization and everything, it's good that we continue make sure that our herd immunity is achieved by giving additional vaccine after that. So for information also in NIH, there is 2 study post vaccination. First among the healthcare workers, that one led by the Institute Clinical Research. They follow up all the healthcare workers after vaccination for 2 years to monitor all the immune system. And then for the post vaccination after among the community, we have many vaccines, right? We have 6 vaccines now. So all that will be monitored by our team also in NIH. I'm also involved in that project. The lead project for that post vaccination among community led by the Institute for Public Health, IKU. So hopefully by all this information, all this data, we can really answer whether or not to give the vaccine after 1 year or not, Prof. From this
Okay.
data. So at the moment, we just say based on our overseas data, right? Even now in overseas, they want to introduce third booster, but I think all over the world now, they want to make sure that 80% of their population vaccinated. Even US not reach 80%, Prof. US still not reach 80%.
Yeah.
So now the main target now not only to say about the booster. For me now in Malaysia, the main target now ensure that 80% of our population be vaccinated.
Yeah, at least the
Hopefully.
first dose, yeah.
Yes, betul. Yes.
Okay. I have another few things which are quite related to immunology, but not to vaccine, perhaps.
Okay.
Now, I think many people now are interested about the, RTK saliva
Okay.
antigen test. You see?
All right.
You have any idea or any information about, RTK antigen test saliva, whether is approved already by the MOH or IMR or how?
Yes, Prof. Actually, for information, I'm also actually, Prof. The one that you asked really suitable for me also because I'm also one of the technical experts in Medical Device Authority (MDA), where the MDA actually body under KKM also, but it's a what call separate entity who actually make sure that all the kits, the test kits, either the PCR, the antibody test, the saliva test, must be tested and evaluated to see that the sensitivity and specificity achieve certain standard. So to answer your question about the saliva home test kit, you will want to know about the home test kit, right?
Yeah.
It's actually we technical experts in MDA, myself and other colleagues in from MOH and also university, we review, Prof. All these first, there are 2 layers. First, we review their data, the company data, their evaluation, their performance. If their evaluation performance not reach to our standard, so we cannot accept. We just say that you must improve your kit. Once they bring back and they say that their data is really good, then we do the evaluation. So who will do the evaluation? Either IMR, either Makmal Kesihatan Awam Kebangsaan Sungai Buloh, or the universities and even government hospital. So from this, we do our own evaluation. Then we get the result. So the one that already stated in MOH, actually the one that already passed all this requirement. So please use the one that listed in the MDA website. Any test kit COVID-19, I hope that everybody can just go to the website of the MDA and follow the list of the kit that already listed there. Don't want, I advise not to try to bring in something that not listed in there, because that list actually very comprehensive and good for the people to actually refer.
Okay.
So it is reliable, Prof. The one that listed by the MOH is reliable lah. Can
Okay.
use. But the problem,
Betul.
Prof,
Yeah.
because it's
Yeah.
home test kit, I
Yeah.
hope people can really understand how to use it. For example, sometimes people can just put a little bit saliva and then they thought it's negative. But actually the amount of saliva not enough, something like that. So they must follow the instruction clearly.
Agree.
Some kids just give 12 steps. Some kids just give 5 steps. So depending. I hope people can just follow the instruction one by one. It's not like urine pregnancy test, very easy. It just
Yeah.
not really a difficult step. Urine pregnancy test very easy. But for the saliva test, they need to add the reagent whatsoever. It's quite difficult lah. It's quite difficult. Yeah.
But in case they have doubt about it, so they have to perhaps do the PCR test, right?
Yes, the PCR of course, because the healthcare workers will take the swab, not themselves. That's
Another one, Dr. Masita, is about the Malaysian-owned manufactured vaccine.
okay. All right. Okay. You want to ask whether or not we have, right?
Yeah.
Okay. Prof, you speak to the right person lah. Actually, I'm also involved in this vaccine development for COVID-19. Okay, the history is like this. Before this, Prof, in Malaysia, we don't have this vaccine manufacturer lah, to be honest.
Yeah.
Because why? Because before this, we can purchase the vaccine easily, only for the childhood immunization. So during the starting the outbreak last year, in April, MOH, NIH, all the research institutes in NIH and also the universities, we sit together in April 2020 and discuss. We need to research for vaccine also. So the one that actually involved in this vaccine is two group from the IMR, my team, myself and also Ching Yiming. We both of us do the development for mRNA vaccine. And also for another group in IMR, Dr. Rozainani, Dr. Siti Norzawani, both of them using the inactivated viral vaccine. Why? Because in the virology unit in IMR, we have the BSL-3. We culture, we grow the virus in the IMR. So we can do the technology viral inactivated viral virus vaccine. So these two projects, and then the universities also, they have their own project. They want still under POC, proof of concept. Once we already have the proof of concept, we must do the preclinical. Preclinical means the animal study. The animal study, we must do two type of animal. The small animal, the small animal is okay, then we proceed with the big animal, the monkeys, the primates. The problem now, Prof, in Malaysia, we only have the small animal facilities. The big animal, we don't have in Malaysia. We have to go overseas, the nearest Thailand. So after we pass the preclinical, we must do the clinical trial. Clinical trial, I think is no problem because Institute Clinical Research in NIH very well versed in clinical trial. No problem. So in terms of the manufacturer, now the one that already have only for Pharmaniaga using the fill and finish products. Means that they bring the bulk from the Sinovac and they just bottle here. The bottle here not very simple, Prof. They have 13 QC need to be passed to be up to the standard by the NPRA. Then can be used by our Malaysian. So Pharmaniaga, and then for our product, our vaccines later on, we still negotiating with the MIDA, the Malaysian Development Agency, to find the suitable companies to produce our vaccines. But the moment still at the proof of concept, Prof.
Yeah. Thank you, Dr. Masita.
Okay.
I think there is no other questions, right? You have questions, huh?
We have questions from the audience.
Okay.
Nampak?
Question to Dr. Masita, can you comment on the need for third dose already, kan?
Okay.
Okay. Is there any current research for Sinovac on Delta variant? I think it's answered already. Uh, uh, Question for Dr. Masita semua. Can you comment on the idea of mixing the various type of COVID-19 vaccine?
Okay. At the moment, Malaysia, we still waiting for the WHO lah. Once the WHO recommend that we can have the mixing vaccines, then we can follow. Another thing, overseas in UK, they already publish their data. What they do in their study, they give the 2 doses of Pfizer, and then after 6 months, they give the booster. Booster means the to give another vaccine, the 3rd dose, AstraZeneca. And they showed that the immune system, the level of antibodies increase very good lah, highly increase. So that's the study from 3rd dose from mixing vaccine in UK lah, the one that just published. But still only one study lah. So we need to even in Thailand also they have started already mixing, I know. But for us in Malaysia, we must follow WHO once. And if really we want to do this mixing vaccines, we have to do our own clinical trial. If WHO didn't recommend, we need to do our own clinical trial. At the moment, our top management still try to analyze the data first before deciding all this mixing type of vaccines. Okay, I hope I answered that question.
Okay, I think the next question is already answered. What is the maximum period to wait before taking the second dose? You mentioned that one. Yeah. Especially AstraZeneca, Sinovac, and this one. What to do if exceeding that period? What is the last update about mixing vaccine? Already answered, and what is the practice? Okay. Next one, for people who are not sure they actually receive the vaccine because of empty syringe, what do you propose? They are not sure.
Not sure,
They are not sure.
Another one thing, of course, they can do the blood test lah. Blood test, because blood test easier to check. So they can check the antibody level of the COVID-19. But the type of antibody must remember to spike protein. Spike protein. Because antibody also can detect nucleocapsid. So more all vaccines actually towards the spike protein. If you remember just now my presentation during the mechanism, all towards the spike protein. Even the Pfizer mRNA towards the spike protein. Even the Sinovac also towards the spike protein. Even for the AstraZeneca also the spike protein. So make sure the type of antibody very important. If you do the blood test for antibody towards the nucleocapsid, you said, see, no antibody what? Empty shrink. But actually, you actually test to the wrong antibodies lah. So please remember, if you really want to do the blood test, please ensure to do it towards the spike protein. That's another thing lah. Secondly, we can do also neutralizing antibody lah. Neutralizing antibody also can be done for the to ensure that really or not you have received the vaccines or not. So to answer the question from Dr. Teh here, use the antibody towards the protein.
Okay, Dr. Masita. Okay, I think time's already up for us to continue the discussion. It's very interesting for me because I think I learned a lot from you. Okay, so I think with that, I think we all appreciate your involvement in this conference. Thank you very much, and we would like to end the session with wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi taala wabarakatuh.
Waalaikumsalam.
Thank you very much, Dr. Masita, Arip.